ladies and gentlemen. I would like to welcome you to our online lessons. This is Chemicals of Life, and we are in lesson 4. And in this lesson, we are looking at some of the carbohydrates, and we have called them monosaccharides. So we'll find out what monosaccharides mean shortly. But I want to welcome you once again, and I want to encourage you to subscribe to this YouTube channel for more information like this. So you can see some of the pictures there of some of the most commonly found monosaccharides: glucose, fructose, and galactose. There, those are some of the common monosaccharides that you can find. So, like we said earlier, that the most frequent monosaccharides are the hexose sugars, which include glucose, fructose, galactose. Those are the hexose sugars. We're going to find out what hexose sugars mean. So these are sugars basically with molecular formula C6H12O6. Now, C6 means there are 6 carbon atoms, and that's where the name hexose comes from. From the words like hexa, hexa meaning 6. You remember hexagon, a 6-sided figure or a 6-sided structure is called a hexagon. Yes, you say hexagonal shape, it means the shape is 6-sided. So hexose sugars are sugars that have 6 carbon atoms, and the structure, the molecular formula is C6H12O6, meaning there are 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. The structural formulae can be shown next, but you know the structures may vary from the molecule. So we're going to see shortly that sometimes the structural formulae are slightly different from the molecular formulae. Yes, we can see one of the structures there of the hexose sugar, and we want to call it aldose sugar. From the word aldehyde, the word aldose comes from the word aldehyde. So aldehyde means an aldehyde is a carbonyl compound. You will see this in organic chemistry. It's a carbonyl compound that contains, of course, carbonyl compounds contain a functional group C double bond O. I want you to look for where that is in that structure. C double bond O. That one is a carbonyl compound group. Yeah, carbonyl group, functional group for carbonyl compounds. So when that C double bond O is found at the terminal end of the structure, if it is located, if its position is, or its location is at the end, yeah, meaning there is no any other carbon atom beyond the carbon atom with the double bond, that one is an aldehyde. And because it is a sugar, and you know sugars end with the suffix "ose," that's why we call them aldose, from aldehyde and the suffix "ose" of sugars. So they become aldose sugar. So it means that a sugar that has an aldehyde functional group. So aldose sugar is that one there. How many carbon atoms are those? 6. And there are how many hydrogen atoms? 12. And how many oxygen atoms? 6. So it is a hexose sugar, but because of the functional group located at the end, it becomes an aldose sugar, or aldose hexose sugar. Let's see another one. This other one here is called ketose sugar. So a ketose sugar now, from the word ketone or ketone. A ketone is also a carbonyl compound. It bears the carbonyl functional group C double bond O. But unlike the aldose sugar, the ketones have the C double bond O in between the carbon atoms. So the C is located between 2 other carbon atoms. So it is in the middle of somewhere where there are carbon atoms surrounding it. So that one becomes a ketone. You remember an aldehyde, the C double bond O is at the end of the structure, And then in the ketone, the C double bond O is in the middle. So a ketone, which is a sugar, is called a ketose sugar, as you can see there. So we have a classification of hexose sugars basing on their position of the functional group. They can either be classified as aldose sugars or ketose sugars. I want you to search for more examples of aldehydes and more examples of ketones that are sugars, and then you can send me some of those examples. I'll be glad to see your responses. That is your assignment number 1. Okay, let's see now the comparison here. Let's look at glucose and fructose. You see that glucose here in this case is having an aldose structure. As you can see, C double bond O is at the end. What about fructose? Have you seen where the C double bond O is located? Yes, it's located somewhere in the middle where it is sandwiched between 2 carbon atoms. Therefore, it qualifies to be a ketone, and therefore it will be ketose sugar. So we have the glucose, which is the aldose sugar, and fructose, which is a ketose sugar for this case. So I want to task you to learn how to differentiate between the ketones, the ketose sugars, and the aldose sugars. That will be very great for you to learn that, and very interesting. Thank you very much. Let's proceed to look at what we call isomerism in monosaccharides. You remember the word isomerism in your O level, or isomers. This is, of course, you remember we defined it in chemistry as existence of molecules, one or more than two structures that are derived from one molecular formula. Yes, meaning one molecular formula can terminate into more than two or more structural formulae. You can find the same compounds having the same molecular formula but having a different structural formulae. So that one is called isomerism. So even in chemicals of life, isomerism is not spared. There are some molecules that conduct isomerism. There are some compounds that exhibit isomerism. One of them is glucose, and we know that glucose can exist in a number of isomers where it has different structural formulae but the same molecular formula. You remember that the molecular formula for glucose is C6H12O6, but when you look at the structural formulae, they may be different. Yeah, that is C6H12O6 has a number of structures that it can produce, and that is what we call isomerism. An example is this. Glucose has a number of isomers. One of them is called beta glucose, and I want you to see the colored OH. Let's number those carbon atoms. Carbon atom 1, 2, 3, 4, 5, and 6. And let's look at carbon atom 1 for now. And you're able to see that there is OH in carbon atom 1 and also H. I want you to look at the position of OH. OH is positioned up above the carbon atom, above the hydrogen atom. That one is what we call beta glucose. And then when you look at this one here, let's look at the position of OH from carbon atom 1. You realize that the OH is located below the H. And of course, that definitely means we have not removed any atom from this structure, but the position changed. Position of the atoms have changed, and that definitely becomes a different structure. So from the same molecular formula of C6H12O6, we now have two structures. The first one there, where the OH is above, is called beta glucose, and then this one here is called alpha glucose. So beta glucose and alpha glucose are some of the isomers, are all isomers of glucose. they all have the same structural, same molecular formula, but they exhibit isomerism and they produce different structural formulae. That's great. Let's see much more clearly here. You can see the position of the OH in both alpha glucose and beta glucose. You'd see that the OH position is different. In alpha glucose, the position is below the hydrogen, and then in the beta glucose, the OH is above the hydrogen. So those are the two isomers of glucose that we are looking at here. Therefore, glucose exhibits isomerism. Glucose exhibits isomerism. Great. Let's proceed to look at the structures of hexoses much more. You realize that we remember what we said about hexose sugars. These are sugars that have 6 carbon atoms. And when you look at their structures, you realize that they can also exist in a straight chain, a straight or in chain form. So you can say it exists in a straight or chain form. And then they can also exist in a ring form. We were going to see what a straight or chain form means and what ring form. So we're going to look at them here. So let's look at the pentose sugar. Before we are going to see shortly what a straight and ring forms mean. What you are seeing here is a ring form, and what we saw earlier is what we call the straight chain. This one here is a straight or chain form of glucose, for that matter, or for fructose. And then what we are seeing here is a ring form. Those are ring forms. So I want you to learn how to differentiate between a chain form and a ring form as structure. So these sugars have different structures. They can exist in chain form. They can also exist in ring form. We are going to continue interacting to discuss more about chains and rings as we proceed. But for now, let's look at pentose sugars. From the word penta, you remember the mathematical pentagon, a 5-sided figure. So if we say pentose, we're simply meaning a sugar molecule that has 5 carbon atoms. So that is a pentose sugar. So they are found in nature as they exist as ribose sugars and deoxyribose sugars. And they also exist both in straight chain and ring forms, as we shall see now. So when you look at this one here, you are seeing a straight or chain forms of pentose sugars. So ribose, straight chain of ribose sugar looks like that, and a straight chain of deoxyribose also looks like that, as you can see from those figures there. So how do we know that they are pentose sugars? We count the number of carbon atoms. So you realize that in a ribose there are 1, 2, 3, 4, 5 carbon atoms, meaning it's a pentose sugar. What about the deoxyribose? Let's find out. Let's count together. You realize that there's 1, 2, 3, 4, 5. So there are 5 carbon atoms also, meaning that those two are pentose sugars, and they are chain forms of pentose sugars, as you can see. And then the ring form is that they form rings. Yeah, so you can see from there the deoxyribose and then the ribose sugars. The green one is the ribose pentose sugar, and then the blue one is the deoxyribose pentose sugar. So I want us to differentiate between ribose and deoxyribose. Let's observe the two forms, two rings, and then you tell me the difference. What have you seen? They look almost the same, but you realize that if for the ribose sugar, look at the OH there at carbon atom 2, and then look at the OH, look at the carbon atom 2 at the deoxyribose. You realize that there is a difference. You see that for the ribose there is OH, And then for the deoxyribose, there is only H, meaning one O is missing in the deoxyribose sugar. So that's the difference between those two, is the number of oxygen atoms. Deoxyribose is less by 1. So that is the difference that you can see from those two. That is great. So ribose, where do we get the ribose sugars naturally? Where do they exist? So you can get them, they act as coenzymes in ATP, adenosine triphosphate, and they are also constituents of RNA, ribonucleic acid. What about the deoxyribose? Deoxyribose is a major component sugar of the DNA, the deoxyribonucleic acid. So RNA has the ribose sugar, RNA and ATP. The sugar that they have is called the ribose sugar, ribose pentose sugar, and then the DNA contains the deoxyribose sugar. That's why they are called deoxyribonucleic acid, DNA. So naturally, you can be asked where those two pentose sugars exist. Where can you get the ribose sugar? And then where can you get the deoxyribose sugar? Where is it found in living systems? Great. Let's look at another category of sugars that are called the triose sugars. So from the word tri means 3. So if we say triose sugars, what do we mean? We mean that they contain 3 carbon atoms. Yes. Remember we had hexose sugars that contain 6 carbon atoms, pentose sugars that contain 5 carbon atoms, and now we are talking about triose sugars. These ones contain 3 carbon atoms. And the two popularly known triose sugars are the glyceraldehyde and dihydroxyacetone. So those are the two commonly found triose sugars, glyceraldehyde and dihydroxyacetone. So both of them are found in plants and animals, and they play a role in carbohydrate metabolism. So you can see there the different structures of the triose sugars. The glyceraldehyde, the chain form, and the hydroxyacetone also, the chain form looks like that. They all have 3 carbon atoms. And then you also look at the ring forms there, the rings of both. You'll see that they are slightly different. So those are the structures. So generally, looking at monosaccharides, those are the structures of monosaccharides. And we remember we classified the monosaccharides as, first of all, depending on the number of carbon atoms, they can be classified as triose sugars, those ones with 3 carbon atoms, pentose sugars, those with 5 carbon atoms, and hexose sugars, those with 6 carbon atoms. And then we can also classify them based on whether they are aldehydes or ketones, depending on where the functional group C double bond O is located. We can also classify them as aldose sugars and then as ketose sugars. As you can see from that summary there, I would want you to clearly identify the triose sugars, the pentose sugars, and hexose sugars, and also identify the aldoses and then the ketoses, and then explain why they belong to those two groups. So I'm giving you 1 minute to do that, and then you summarize in your book. Yeah, so we can proceed. We can also look at the disaccharides. Remember, we have looked at the monosaccharides, where we said that monosaccharides are majorly are sugars that contain just one sugar molecule, and they are very simple sugars. So this time we want to look at the disaccharides. That means two, and if we say disaccharides, saccharide means sugar. So there are two sugar units combined together to form disaccharides. So generally, monosaccharides combine together in pairs to form disaccharides. This union involves loss of water molecule, and therefore the reaction is a condensation reaction. Remember what a condensation reaction means? A reaction between two simple molecules to form a complex molecule with the loss of a water molecule. So that one is what we call condensation reaction. So a condensation reaction takes place when two monosaccharides combine to form a disaccharide. A disaccharide plus a water molecule. So, and the bond which is formed between the two monosaccharides when a disaccharide is formed is called a glycosidic bond. Yes, so what I want you to note from these two paragraphs is that what is the name of the reaction that takes place? It's called the condensation reaction. And then what is the name of the bond? The bond is the glycosidic bond. So the common examples of disaccharides, the most common ones, are maltose, sucrose, and lactose. And maltose is formed when two glucose molecules combine. Glucose plus glucose, you form maltose. And usually we find maltose in germinating seeds and in cereals. And then sucrose is formed when we combine glucose and fructose. And naturally it occurs in sugar canes. And then we have lactose, which results from the combination between glucose and galactose. And usually we find it in milk. So those are the three commonly known disaccharides. We have maltose, sucrose, and lactose. So let's look at their properties very fast here. One, they are sweeter than monosaccharides. So these disaccharides are much more sweeter. If you bought glucose from the shop and then you test it, and then you also bought sugar from the shop, which is sucrose, and then you test, you may realize that the sugar, the table sugar at home, might be slightly sweeter than glucose. I want you to try this out and see whether it is true. And then two, they can be crystallized. They can form crystals. And then they are soluble in water. When you mix your sugar with water, you form tea. With hot water, you form tea. It's because they are soluble in water. And then they do not change the color of Benedict's solution when heated with it, apart from one which is called maltose. So they are most all of them are non-reducing sugars apart from maltose. We are going to see what non-reducing sugars are or what reducing sugars are shortly. And then they can also be broken down into simple sugars by dilute mineral acids and enzymes. So if you treat them with dilute mineral acids like nitric acid or hydrochloric acid, these disaccharides can be broken down to monosaccharides. Also, if you treat them with enzymes, for example, if you treat sucrose with the sucrase enzymes under suitable conditions, sucrose will be broken down to glucose and fructose. If you treated lactose with the lactase enzyme, then lactose will be broken down to galactose and glucose, and so on and so forth. So they can be broken down into simple sugars under action of enzymes or acids. And then maltose can be formed as when we break down starch during hydrolysis, for example, during digestion in our bodies, in the mouth. Let's look at there the formation of maltose. You can see two glucose molecules, C6H12O6 plus another C6H12O6 to form C12H22O11 plus water. So that C12H22O11 is maltose. And when you look at the structure of the reaction there down, you can see glucose plus glucose. The blue rings are the OH and the H combine to form water, and then the bond forms between carbon atom 4 and carbon atom 1 there. Then you form what we call 1,4 glycosidic bond, the bond between carbon atom 4 of one glucose molecule and carbon atom 1 of another glucose molecule when water is lost. That bond which forms there is called the 1,4 glycosidic bond, and then you have maltose there. So that's great. And then we also have formation of sucrose in the same way, where glucose and fructose combine, and then sucrose is also formed. In the same way, all the two water molecule is lost and a bigger molecule is produced, and that is what we call condensation reaction. So I want to thank you very much for being very good students and following these lessons online. In case you've liked our lesson, you can subscribe to this YouTube channel and then like and share to other students as well. Please continue staying at home. We wish you the best.